Hey everybody, we're gonna start our section number 4, Back to Basics, Vectors and Vector Control in the Context of a Dengue Endgame. And it's a pleasure to be here. I'd like to thank the organizers for allowing me to moderate this section. And our first speaker is Jason Rasgon from the Pennsylvania State University. Jason, you have 20 minutes. Thank you.
Okay. How do we move the slides? So,
I'm guessing it's the green one.
okay. Okay. So I'd like to thank the organizers and Adam for inviting me to this, my first time at this meeting. It's been really good. So today I'm actually going to sort of take a summary of some of the work my lab's been doing for about the last 15 years. So we're kind of going to go all over the place. And I apologize in advance. This is a dengue meeting. My lab until very recently didn't really work on dengue. So a lot of the stuff I'm going to show you, while it's relevant to the topics, is actually not on dengue. We'll hit dengue towards the end because it's sort of the later stuff where we actually started to work on that. I'd just like to thank the many different enablers over the years that have allowed us to do this research. A lot of it primarily funded by the National Institutes of Health, Allergy and Infectious Diseases, also Pebble Labs, and significant resources from Penn State and the Huck Institutes of the Life Sciences. And also, I don't do any of this work. I sit in my office and type. These people do the work. And so this is not all of them. I couldn't fit everybody on the slide, but I do want to specifically highlight the people in red here. A lot of this work started with Brittany and Grant and a lot of the other people. And everybody else in the lab has had various sort of supportive roles here and there over the years. So we've heard a fair amount about Wolbachia during this meeting, and I think we're going to hear some more after I talk. So this is my one slide on the World Mosquito Program. We all know World Mosquito Program. We know the sort of the Wolbachia strategy. I just thought it was interesting. Every time I talk about this slide gets updated. These are the data that I actually saw as of current, I think of last night. So they've released Wolbachia-infected Aedes aegypti. Is that working? Yeah. In 42 field sites across 15 countries so far. So that's pretty phenomenal. But here's what I want to talk about. So in some sites, and we've seen this, Wolbachia invasions into natural populations have been successful. They've increased in frequency over time. They seem to be stable, and we saw some of that data earlier. And we see significant decreases in dengue virus incidence, which is a good thing. There's been a successful randomized control trial in Indonesia. There are other ones ongoing and also planned, and so these things are being significantly advanced every single year. But there are sites where the invasions have been difficult. They release Wolbachia, they don't spread, they get lost. And pathogen decrease has been unclear in some of these areas, even when Wolbachia reaches fairly high frequency. There seem to be significant, for instance, heterogeneity in Brazil and Colombia and other places. And so there are some issues here. We don't even know really what the primary mechanism is by which Wolbachia modulates or blocks pathogen transmission in Aedes aegypti or mosquitoes. There are a lot of hypotheses for this, that Wolbachia induces immunity in the mosquito, which downregulates virus, or resource competition between Wolbachia and viruses, primarily for lipids and cholesterol. There's some evidence from microRNA modulation or RNAi modulation. But even though we don't know this, it's still kind of a black box. Releases are being conducted anyways. And this is a problem because when something goes wrong and you don't understand how it's working in the first place, you don't know how to deal with it or mitigate those problems. And something I've been hitting for about 15 years now, Wolbachia does not always suppress pathogens in mosquitoes, or really even in other kinds of insects. This is a common phenomenon. We see sometimes enhancement of pathogen infection or transmission. We see different effects on different pathogens. Sometimes it blocks one pathogen and suppresses or enhances another one. There can be significant variation due to environment. We heard a little bit about temperature. WML doesn't seem to like really hot temperatures. And then lab studies, it's actually pretty difficult sometimes to detect instances where Wolbachia can fail. It's hard to sometimes power experiments, for instance, to see these kind of phenomenon in a statistically significant fashion. So the questions that we've been interested in for a long time, how much variation do you see in pathogen suppression phenotypes by Wolbachia? And this turns out to be dependent on a lot of things. Mosquitoes, mosquito genotypes, viruses, which viruses you're talking about, what viral genotype, what Wolbachia strain, what are the environmental conditions and parameters that govern this? And what are the mechanisms of Wolbachia pathogen effects? And there's a typo, though, that I did not catch. You always see it when you're actually talking. And then what are the implications of these for releases? So I'm going to run through some data, about 15 years worth of data. I'm going to kind of go all over the place, just to give you a flavor for the variation that we can see. So in these data here, this is on bioRxiv, it's in revision, where we actually artificially infected Anopheles gambia and Anopheles stevensii with Wolbachia, both with WLB and WMEL. We infected them with Onyongyong virus, which is an alpha virus that infects Anopheles. And we see very good suppression. So the laser pointer doesn't really show here, but you can see both in Gambia and Anopheles stevensii, both LB and WMEL seem to significantly suppress viral titers. This seems to be very, very similar to what you see with Aedes aegypti and dengue virus. Not too surprising, maybe. And this actually is due to enhanced toll signaling. So the Wolbachia infection upregulates toll in the mosquito, which downregulates virus. And I'm not going to go through all the mechanisms of that because I don't have time for that, but it's on bioRxiv if you're interested. Also, we artificially infected a different mosquito, Culex tarsalis, which is a native North American vector. And we see that it suppresses Rift Valley fever virus titers. This was done in collaboration with Mike Terrell. And again, this is two different day 7 and day 14. This was with WLB, and we see significant negative correlations. The higher the Wolbachia levels are in the mosquitoes, the lower the viral titers. Again, fairly canonical virus blocking. But about more than a decade ago, we saw this, where we also infected Culex tarsalis, and we challenged them with West Nile. And it turns out when you infect these mosquitoes with, this is WLB, they become about twice as likely to become infected with West Nile. So it is not suppressing virus. And actually, we don't see any variation in titers. And so this was quite interesting. And this is sort of where we got onto this whole idea of variation. I'll talk more about that later when we get into some mechanisms. Here's another paper we just published very recently where we infected Aedes aegypti, and we tested them with different alpha viruses. This is Synbis virus, and both LB and WMEL increase infection prevalence, at least at day 7. And also we have significant increase in titer from LB at day 7. So the Wolbachia-infected mosquitoes actually have higher virus than the controls. What's going on? This is myerovirus in Aedes aegypti. WMEL, we see almost perfect blocking. And again, there's another paper published before we did this, actually. They saw basically the same thing. So WMEL seems to work very good against myerovirus. But when we looked at WLB, and this is actually even with different titers of Wolbachia, we see very poor blocking. And I'm going to summarize a whole lot of data here from many, many different studies just to show that there's a lot of variation here. So sometimes, depending on the Wolbachia strain, the mosquito, and the virus, you have blocking. So we can see here, we see blocking of Bunya viruses, Rift Valley fever. We see blocking of various alpha viruses. We see blocking of other kinds of things. Sometimes you don't see any effect, and I didn't talk about some of this, but where we see no real effect on Synbis virus in tarsalis, which I didn't talk about. We see the same thing with Aedes aegypti and Onyongyong virus. Or we can see enhancement. So West Nile, Synbis virus and Aedes aegypti, we see some other things. So there's a lot of variation happening here. It's not always just you infect the mosquitoes and it blocks virus. We even see this with Plasmodium. So this is actually combined data from a number of studies where we see both LB and WMEL or WMEL pop block Plasmodium falciparum. We see LB actually enhances Plasmodium berghei, however, which is a rodent malaria, whereas MEL-POP actually suppresses it. And Plasmodium yoellii, which is rat malaria, we actually see enhancement by both LB and WMEL. And there's a temperature component here, which I'll come back to in a minute. So we can see the full gamut of variation also with parasites, not just with viruses. So where are our breakdown points? Is there resistance to invasion or pathogen blocking due to environmental variables, human activity, mosquito host genetics, temperature, insecticide resistance maybe? There was some data to show that insecticide resistance may actually be stopping originally the spread of Wolbachia in some Brazilian releases. Microbiome effects, which I will show some data on. There can be potentially selection on the viruses to overcome Wolbachia blocking. RNA viruses have very high mutation rates, so this is a selective, they will respond very quickly to selective forces. There can also be selection on Wolbachia to attenuate blocking, particularly if blocking is correlated with fitness effects in the host. Yes, so high Wolbachia titers cause strong blocking, but also causes fitness effects. So you select actually for lower titers at a population level, which selects independently for lower blocking potential. And there can be selection on the mosquito host to attenuate blocking. And I'm not really even going to talk about fitness effects in this talk, because that could be a whole other talk. I'm really going to focus just on pathogen blocking. So here's some temperature data. This was where we infected Anopheles with Al-B, and we reared them at different temperatures, and then challenged with Plasmodium yoellii. And depending on what temperature we reared the mosquitoes at, we could see different phenotypes. And we had two different controls here, a unmanipulated control and mosquitoes that were injected with cell culture lysate. At 20 degrees C, basically, we don't see enhancement or blocking. At 24 degrees C, we see significant enhancement in Plasmodium oocyte levels in the Wolbachia-infected treatments. But at 28 degrees C, we see the opposite. We see blocking. So we have not done these experiments with viruses yet, just with parasites. I would be shocked if we don't see something similar, because temperature changes a whole load of things in the mosquito. Microbiome. I talked about how Wolbachia infection actually increases susceptibility of Culex dorsalis to West Nile virus. It turns out it's because there's some third order effects happening here. When you actually infect the mosquitoes with Wolbachia, we see significant downregulation, so the green, of a group of bacteria called Acetobacteriaceae. It actually turns out a specific one, it's a bacterium called Acaya. So Wolbachia and Acaya actually have negative interactions on each other, and they're reciprocal. And we showed this in other contexts many, many years ago, and other groups have seen this too. Turns out Acaya actually confers resistance to West Nile. So just showing here, when we infect the mosquitoes with Wolbachia, in Aegypti, there's no significant relationship, but in Tarsalis, there's a negative correlation. So the higher the Acaya levels are, the lower the Wolbachia, and vice versa. And Acaya actually reduces West Nile. So when you can just challenge and load mosquitoes up with Acaya and challenge them with West Nile, we see basically about a 60% reduction in the infection rate. It's a similar magnitude to what we saw in the opposite direction with the Wolbachia challenges. So the model here is that Wolbachia infection reduces Acaya levels, which increases susceptibility to virus. Now, whether this happens in the field, I don't know. This is a lab study, but it does say that the microbiome can actually have significant effects on this interaction. I'm going to finish here with host effects. So is there selection on the mosquito host? So this is some data from a colleague of mine, Beth McGraw. They did a study a few years ago where they actually took Wolbachia-infected mosquitoes out of the field in Australia and then challenged them with dengue virus, and then they selected them for mosquito lines that were good blockers. So they're all infected with Wolbachia, but they selected for ones that were good blockers and ones that were bad blockers. And then they did a random selection as a control. This is published a few years ago now. So you've got high blockers, low blockers, and random blockers. And they only did this for 4 generations, so not for very long at all. And they actually were able to show this, where they could actually select for Wolbachia-infected mosquitoes that were bad, they were very poor at blocking dengue virus, or very good at blocking dengue virus. The randoms were kind of somewhere in between. Now they have this, Beth is an evolutionary geneticist, and so they did this big genetic screen, and they found candidate genes in the mosquito genome that were responsible for mediating, or at least correlated with this interaction. There were about 60 of them, but this peak here on chromosome 1 stood out. So there are about 60 associated genes, and there are actually 2 genes in that big peak. It's alpha-monosidase 2A, and then cadherin 87A. So in this paper by Beth and Susie Ford, actually was the first author, these genes are not immune genes. They're associated with like glycosylation and cell adhesion, things like that. There was some data to suggest that, at least for alpha, I'm not showing the cadherin data, but I'm showing the alpha minosidase data, that this might be involved in the dengue infection phenotype. So in Wolbachia uninfected mosquitoes, they did RNAi and knocked these down. And when you knock down alpha minosidase 2A, the dengue titers go up, and also the dengue prevalence goes up. But again, these are in Wolbachia uninfected mosquitoes. And the problem was, when the mosquitoes were infected with Wolbachia, the RNAi didn't work. So you see the top there, there's no effect on, when they're infected with Wolbachia, there's no effect on the double-stranded RNA treatment. If the mosquitoes don't have Wolbachia, RNAi is very efficient. So this is as far as they got. My lab works on CRISPR. So we started working with Beth on this, and we knocked these genes out. We actually knocked out cadherin too as well, although we lost that line during COVID before we could characterize it. But we were able to do the alpha minosidase too. So I'm not going to go through this. We established a homozygous deletion mutant line for this gene. It was viable. It wasn't particularly happy, but we could rear it. It's just a mutation. Actually, in this allele, actually the same allele has two different knockouts. It causes about, I forget what it is, 50 nucleotide differences, a large deletion and another smaller deletion. We can screen for these by PCR. That's how we did it. And then we introgressed Wolbachia into the line because we didn't want to do the knockout in the Wolbachia line. We didn't know how Wolbachia would affect the CRISPR. We were able to outcross it and then essentially homogenize it with the control line. We got this. This is a W-ALB we got from Zhongzi at Michigan. So now we have Wolbachia-infected and uninfected mutant wild-type mosquitoes. Now, the RNA, or in Susie's paper, there was a little bit of a suggestion to say that the knockdown mosquitoes might have higher Wolbachia titers. We confirmed this. So the knockout mosquitoes actually have about, on average, twice the Wolbachia load that the controls do. So you would think, hey, they should be better blockers. Not necessarily. So we did the challenges. We have, again, Wolbachia positive, negative, and then wild-type mosquitoes and homozygous knockout mosquitoes. We fed these with an artificial blood meal, either on dengue too. I said I'd get to some dengue data, so here we go, or myarovirus, which is sort of our lab house virus that we use a lot. And then we looked at infection, dissemination, and transmission by saliva. And my lab always does these with live virus assays. We don't use qPCR. So everything we were here is live viable virus. Okay, that didn't work great, but I'll just do this. So the Wolbachia negative wild-type mosquitoes, you can see that in the first column. This is for dengue. If you have Wolbachia in the mix in a wild-type background, we see a reduction in virus. I mean, that's what you would expect. The Wolbachia negative knockout mosquitoes also have a reduction in virus, very similar. But when you add Wolbachia into the mix with the knockouts, you completely lose blocking. So there's this interesting sort of interaction. If you just compare the Wolbachia-infected mosquitoes, either wild-type or knockout, you actually see what looks like enhancement of virus. But what it really is a loss of the blocking phenotype. When you look, though, at an alpha virus, we see something completely different. Again, Wolbachia here, you can see in the first two, we see significant reductions in virus. In the Wolbachia negative knockouts, we see actually higher virus than the wild types. And then when we add Wolbachia in there, we see blocking again. So in this case, for alpha viruses, we see significant differences if Wolbachia is not there. But if Wolbachia is there, we see blocking. It doesn't seem to change anything. So flavivirus versus alpha virus, mutation versus non-mutation, Wolbachia versus not Wolbachia. Everything here is affecting blocking. So I hope I've demonstrated to you that Wolbachia effects on pathogens can be highly variable. And it depends on the Wolbachia strain, the pathogen, the mosquito species, the environment, the microbiome, and host genetics. Probably other things, too, that we haven't looked at. These results are complex. They are not generalizable across systems. You have to look at it independently. And there's significant standing variation, genetic variation in the mosquito genome that can affect Wolbachia phenotypes. Beth was able to select these lines in 4 generations. And again, you can select for those very, very quickly. The mosquito host genome and the microbiome can have large effects on the penetrance of these blocking phenotypes and the complex interactions. We tested one gene. We've got about 60 more to look at, and to say nothing about the interactions between them. And I think this has important implications for the stability, safety, and efficacy of control strategies which are relying on Wolbachia-based pathogen blocking. And with that, I will end there. Thank you. Take questions.